Flowering plants undergo a unique reproductive process where there are two fertilization events. In order for the double fertilization event to occur, the male gamete, the sperm, must travel from the anther to the embryo sac within the female reproductive organ. The pollen grain contains two main cells. A cell named the tube cell makes up the bulk of the pollen grain, and the sperm cell, which at this stage is known as the generative cell. To reach the embryo sac, a pollen grain must land on the stigma. Once landed, it begins to germinate. The tube cell forms a long structure down the style and into the ovary. The generative cell travels behind the tube cell nucleus. Once near the ovary, it divides by mitosis to produce two haploid sperm cells. The pollen tube reaches the micropyle and releases the sperm cells into the embryo sac. One of the two sperm cells fertilizes the egg cell. This produces a diploid zygote, which will become the embryo. The other sperm cell moves up and fuses with both of the central nuclei, forming a triploid cell. This unusual triploid cell develops into an endosperm and serves as the embryo's food supply during early development. It is only angiosperms, flowering plants, which have this double fertilization characteristic.
Female cones develop two ovules on the upper surface of each cone scale. Each ovule contains haploid megaspores. Male cones produce microspores by meiosis. The microspores develop into pollen grains. Each winged pollen grain is a four-celled male gametophyte. The female gametophyte grows, producing two or more archegonia, each of which contains an egg. During pollination, a wind-borne pollen grain falls near the opening in one of the ovules of the female cone. Each male gametophyte forms a pollen tube that penetrates the tissue of the female gametophyte. When the pollen tube has grown into the archegonium, a sperm cell from the male gametophyte fertilizes the egg. The zygote develops into an embryo and a mature seed is produced.